Hi, today what we're going to do is we're going to talk about Chapter 15, Chromosomal Basis of Inheritance. This is still talking about genetics and heredity and probably in the sense of exceptions to Mendel's rules. You have the notes, and this also, the PowerPoint can be found on Schoology. The first topic we're going to talk about is autosomal traits influenced by sex. So there's a couple of categories. So the first one is sex-limited traits. But I want you to understand this is different than sex-linked traits. This is not necessarily on the X and Y chromosomes. Instead, these are traits that you find on normal chromosomes, but as for how you see their phenotypes, it's highly influenced depending on the person's sex. So if you see sex-limited traits, these are autosomal traits, meaning found on normal chromosomes that are not sex-linked, but they tend to be expressed only in one sex. So meaning both males and females can carry the genes for it, but only it's expressed in one sex. And in this case, in this example, a beard having male beards. So both females and males can have the genes for heavy beards, but you're only going to see heavy beards found in a male and not in a female because it's limited in the sex. Okay. Another one is going to be sex-influenced traits. With sex-influenced traits here, once again, are autosomal genes, so genes not on the X or Y, but on the normal body genes that represent the rest, not the sex chromosomes. And what they're going to see is that, yes, both males and females will have it, but it will be expressed differently in the different sexes. And so this example is early pattern baldness. You can find baldness in both males and females, but the pattern in males is way more significant than the hair loss pattern in females. So some of the reasons why this is sex influenced is because of other hormones that are produced in males that are different from females, like testosterone may kind of exacerbate or increase the expression of those particular traits. Okay, other things that we're going to talk about is when you find disorders that can show when things go wrong. So in this case, this is talking about disorders based on wrong numbers of chromosomes. So remember we had, I think, an FRQ or a question based on, if you have this particular disorder, it's dominant or recessive, does that create extra chromosomes? And the answer is no. Either you're dominant or recessive, homozygous, heterozygous, recessive, etc. That is going to be different than what you're seeing here. When there's a wrong chromosome number, it's because of a process called nondisjunction. We've talked about this process earlier first semester. Here we are taking a looking at it again. So nondisjunction, just think about, remember in meiosis and anaphase, the chromosomes are going to separate. In nondisjunction, those chromosomes fail to separate properly. Now, if you remember, meiosis has two sets of divisions. Meiosis 1 is your first set of division, and meiosis 2 has your second set. And what you're going to see is that there could be different types of nondisjunctions. You could have, as you can see over on the left-hand side over here, nondisjunction occurring when it's in meiosis 1. So in this case, when nondisjunction occurs in meiosis 1, you're going to find at the end, towards the left over here, you're going to see that all 4 gametes are going to be the ones that are going to have the disorder. So over the left here, all 4 gametes have this disorder. So you're going to see 2 of the gametes have an extra chromosome, and the other 2 of the gametes have 1 less chromosome. Now, the second option for nondisjunction, as you can see on the picture on the right-hand side. And over here with this particular nondisjunction scenario, what you find is that you have 2 cells that are going to be normal in the gametes and 2 cells that are going to have different chromosome types. So one of them is going to have 1 extra chromosome and one of them is going to have 1 less chromosome. So this on the right-hand side again is the nondisjunction event that is occurring in meiosis 2 and not meiosis 1. So if you were to ask yourself a question of which of the 2 would cause more of a chance of a disorder. A non-disjunction event in meiosis 1 is going to lead to a higher risk of having abnormal chromosome numbers. Okay. On that note, you actually have terminology for these non-disjunction events. So aneuploidy is a term where you've got, again, a fertilization of gametes where one or more of the gametes had some sort of non-disjunction event. As a result, you're going to find that the offspring with this condition will have an abnormal number of a particular chromosome. This is a very general term, but then what you see is as you go further into the types of aneuploidy, then you're going to see more specific names. So a monosomic zygote is going to be a type of aneuploidy, and in this particular case, this zygote or this individual has only one copy of a particular chromosome. So in this case, 2n equals 1. So if you were going to say a human had 46 chromosomes and they had also a monosomic event, so 46 minus 1 gives you 45 chromosomes in total. So this human that would be monosomic has 45 chromosomes instead of the normal 46. So you can see that in this picture over here. What you find is that one of the sex chromosomes is missing, but yet again, this is going to be a human just with a chromosomal disorder. Now, the second option for another aneuploidy is a trisomic zygote. So in this particular case, what you're going to find is you're going to have 3 copies of a particular chromosome. So in this case, instead of 46, which is our normal, we're going to add an additional chromosome. So these individuals have 47 chromosomes in total. So here's an example over here where you're seeing this is a trisomy 18, where there's one additional chromosome on 18. So again, examples of aneuploidy. Okay. Now, another term we're going to see is going to be a polyploidy. So a polyploidy is where you're talking about not just one additional chromosome, but we're talking about additional sets of chromosomes. So in this particular case, we have triploidy, which is 3 sets of chromosomes, and tetraploidy, which are 4 sets of chromosomes. Now, polyploidy, this whole process can be, again, due to non-disjunction, but you're talking about a really, really large event where maybe it was a problem where all the chromosomes didn't kind of cross over, and as a result, you're going to see that this is pretty severe. This polyploidy can be common in plants, but it's too severe to really be found in animals. Polyploidies, again, are more normal in appearance, especially for plants, than aneuploids. But once again, this is something that is not so common in humans. Okay, oops. As for humans, we do have some non-disjunction events. Again, depending on non-disjunction events, what you're going to see is some of them can be pretty lethal, depending on the type of chromosome or the number of chromosome. If there's too many of, say, chromosome number 1, chances are that that's not a viable offspring. But we do find that there are some in numbers that can be viable. So some common examples would be Down syndrome, which is trisomy 21, an extra on number 21. We also see Klinefelters, which is XXY, which is an extra X, and that's in your sex chromosome. And then you also find Turner's. With Turner syndrome, it's X and O, meaning that there's only one sex chromosome and just a single X. So the question here is, why are aneuploids of sex chromosomes less lethal? Just remember, you've got 2 of the exact same copies, and then thinking about bar bodies, the extra X chromosome will be inactivated, so we only really have one functional X anyways, and so that's why it's less lethal. Okay. Other things we're going to talk about is sometimes you see that there can be other errors in chromosomes, such as wrong chromosome structure. So the question is, how does it happen, and what are some of the results from that? So with wrong chromosome structures, what you're going to see here is that there can be alterations due to some sort of breakage. So in regards to the breakage, what we're saying is maybe in anaphase, when you're supposed to have your chromosomes separate properly, maybe what happens is part of it gets cut off accidentally, or maybe during crossing over in meiosis 1, prophase 1, what you could see is maybe things crossed over improperly. So these can lead to breakages, it can lead to alterations, it can lead to wrong chromosome structures. And here you have 4 examples: deletion, duplication, inversion, and translocation. So they kind of exactly mean what they say. Deletion, you lost a little section. Duplication, you have a repeat. Inversion means it's inverted or kind of flipped. And then translocation means it's located in a different spot. So here are the pictures of the different ones. I'm sorry that it looks kind of funny and all the words aren't quite displaying properly. But looking at A, the top line, you've got deletion. You should have chromosomes or genes on the chromosome, A through H. But on the left, that would be a normal version over here. Oops. Okay, I fixed that. So on the left, you should have A through H. And if you look on the right-hand side on A, deletion, the D got removed. Again, a little bit of removement, a little bit of a deletion. What you would find are some different disorders that can occur from that. For B, the second one down, you've got a duplication. Here, you should have, again, A through H, which is normal. But if you look on the right-hand side, you're going to see B and C got duplicated twice, maybe because of a crossover event. And 2 of the same sets got stuck on one chromosome, and that's duplication. Next, you're going to see for C, the third one, is inversion. So it should read A, B, C, D, E, but then on the right-hand side, you can see that they flipped it over. So you have A, D, C, B, E. So once again, this could be a result from some type of crossover event that went wrong. And then lastly, reciprocal translocation. What that means is you actually have a crossover event, and ideally you should have a crossover event between 2 identical chromosomes, but in this case, they were unidentical. And so you have A and B crossing over with M, N, and O. Okay, what we found is that certain cancers can be from these kind of translocated chromosome events, and they have found with leukemia, you're actually having a crossover between 9 and 12. Again, we don't really always understand why this happens, but at least we're starting to learn a little bit more about what is happening. And then the last little part that we wanted to add, which is a weird exception to genetics, is the idea of genomic imprinting. This is kind of similar to the same effects of like a bar body, where sometimes certain organisms, certain genes, they can also be silenced. And so in this particular case with genomic imprinting, you have certain genes that are silenced by being stamped. And when we say being stamped, it's stamped by a methyl group, methylation, your CH3. So in this case, if you see, you've got 2 alleles. In blue, you have a normal size allele on the top, and then the maternal, which is mutated. In this particular case, the mutated maternal in red was muted, so therefore the mouse is normal size. But on the bottom, the genes are flipped. The paternal side is mutated. The maternal side is normal. But because the maternal side was methylated and muted or not expressed, then the paternal one was expressed, and now you have a dwarf mouse. So again, sometimes with genomic imprinting, you find different alleles can be turned on and off. And so in this particular case, in both scenarios, the maternal had the methyl group attached to it. CH3. And then the other side, same thing as well. And that is why they were mutated. I'm so sorry. This is a little, I'm a little rusty on making these videos, but hopefully that made a little bit of sense. Sorry, I don't quite know how to zoom out, but this is your chapter 15 notes. And if you have any questions, please feel free to email me. Thank you all so much, and hopefully I'll see you soon. Thanks.